Hey guys, I hope you're having a great Tuesday. I'll be with you guys tomorrow. So your assign yourself is the same thing as yesterday. You just need to make sure you get your summaries from the experiments with the diagrams from yesterday's notes. Make sure you have a little diagram for each one. I will be checking those tomorrow. For your do now, you need to go ahead and read the passage and answer the questions. We really need to work on that reading. I felt like I learned so much at my training today, and one of the big things that they know will help AP Bio students is by reading more. So you have this do now, you have 10 minutes, and then it'll be passed in. So make sure you finish it all in 10 minutes. Okay, if you can go ahead and press pause for me, please, sub, that would be great. Thank you. Okay, time is up. Let's go ahead and move on. With that being said, we'll look at the passage tomorrow and look at the answers and see how you did. For now, just go ahead and make sure your name and your period is on your paper, and then we'll go ahead and pass the papers to your right. Now, with that being said, let's go over some announcements. Tomorrow, I'm so excited. We're going to do the Stunted Growth Lab. So you've probably heard Monster drinks are bad for you. Coffee's bad for you. We're going to see, is that really true? And there's that little myth that if you have those things, you won't grow as much or you end up being very short. So we're going to see, is it true or is it a myth? Also, for Catalyst today, Garcia and De Leon, you need to go to your home rooms. And then from your home room, once everyone's inside the room, then we'll go to the RRC and you'll have a time management session. You'll have a time management session with Samantha Salinas. My Unit 3 retesters will do this Wednesday after school. And if you want tutoring for Unit 4 exam, it'll be Thursday since Friday you won't have tutoring. All right, let's go ahead and keep moving on. And celebrations, woo! I found out that Kevin got into Emory. Kevin, awesome! Woo, woo, go seniors, go seniors, go seniors. Look, I got a senior breaking it down for the seniors. Ow, 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 ow. Awesome, awesome, awesome. If you guys, anyone else got accepted, please let me know. Super happy for you, Kevin. Let me know how much you got tomorrow. All right. Let's go ahead and move on with the agenda. So for the agenda, we'll have the do now, which just got picked up, an epic rap battle, notes, cootie catcher, and then you'll get to do a candy DNA at the end because the sub said you did really great today. So let's go ahead and move on. I want to show you guys the epic rap battle between Rosalind Franklin and Watson Crick. I think it is super, super funny. So for the sub, if you can go ahead and just like get off of this video, and then you'll be able to find Rosalind Franklin on the first tap. So I'll let you guys go ahead and watch that. It is so good. It's so funny. It's like nerdy funny. So I'll let you go ahead and do that, and I'll go ahead and press pause while you watch, and then come back to me, please. Okay, well, welcome back. Wasn't that an awesome song? Let's give it up for Rosalind Franklin. Let's give it up for Rosalind Franklin. I'm definitely going to come back to school like dancing. Like, that's a good song. But enough with that. Let's go ahead and start our notes because we still need to talk about the rest of experiments on discovering DNA. So the first one we're going to talk about is Morgan's fruit fly and Griffith's bacterial transformation experiments. So in Morgan's fruit fly experiments, genetic material is carried on chromosomes. Griffith's bacterial transformation experiments showed that biomolecules from a dead bacteria can actually transfer genetic information to a live bacterial cell. So this meant that there's some sort of information that's being passed on. This is a really big deal at this time. Like, sure, just like I was saying yesterday, you and I, we both know that DNA is the genetic material, but back then, this was a really big deal. They had no idea. Let's go ahead and move on. So let's talk about another experiment. So this experiment was Avery, McLeod, and McCarty's research question. So at the time, scientists agreed that genetic material was either proteins, DNA, or RNA, but a lot of people thought it was proteins. And so these scientists wanted to determine which of these three molecules actually carry genetic information. Of course, like you've learned since 7th grade, the only one that is genetic information is going to be DNA. But back then, we just had no idea. So they went ahead and did this experiment. So if you're wondering what are we looking at, this first one right here is DNA. This one over here, it's single-stranded, so it's RNA, and then we have these nice little like alpha helixes, and we can see these kind of like weird beta-pleated sheets. That's going to actually be a protein, and this is a quaternary structure. Let's go ahead and move on. So let's talk about what experiment they actually performed, because this one it's kind of tricky. They actually used heat to kill pathogenic bacteria. So this is bacteria that is harmful. And then they broke apart the dead bacteria using a blender. They extracted the lipids and carbs because, just like I said yesterday, we know that lipids and carbs, they're not genetic material. This was already like known. So we're going to get those out of there. So we had 3 experimental groups. So the first group, we had proteinases, which means we broke down proteins. And then in the second group, we had ribonucleases, where RNA was broken down. And then the third group, so in other words, our third test tube had deoxyribonuclease, and the DNA was broken down. So we're adding these proteins so that, for example, if I test a bacteria with this, then there's no way that we could see that if proteins were the genetic material, it wouldn't work. If I wanted to know if RNA was the one passing it, I break down the RNA to make sure that it's not the one being passed. And of course, if I added an enzyme that breaks down DNA, then I would know, like if something wasn't passed, then I know that DNA would be the genetic material. You know where this is going to end up. We're going to know that DNA is the genetic material. This is just the evidence on how we got there. So they added each experimental group to a sample of living, harmless bacteria to see if it would be transformed into pathogenic, harmful bacteria. So we've talked about this, where we transform things, where we take something that is not harmful and we transform it to a bad thing by passing on genetic material. So let's go ahead and take a look at the results. So the sample that was treated with proteinases, bacteria were transformed. The sample that was treated with ribonucleases, bacteria was transformed. But when we had a sample with deoxyribonucleases, which is no DNA, we had no transformation. In other words, we took away the genetic information, so it couldn't be passed on to any bacteria. But of course, we know this. We know that DNA will be the genetic material, but this is how they proved it. So what's the significance? The significance is that DNA, not RNA or a protein, is able to transform harmless bacteria into very deadly bacteria. And so this was just proving again, DNA is the genetic material. And so you might be saying, like, I get the point, but you have to know these experiments because on your unit exam, you're going to be given about a page-long reading of an experiment, and you have to understand which scientist was doing it and what they were trying to accomplish. Okay, let's go ahead and move on. So now we're going to talk about how DNA replicates. So right here, what I have is this is the original molecule. Then we have the separation of strands. So these two are the template strands because we're going to go ahead and copy them. And then these parts right here are going to be the new strand. So what I want you to do is I want you to make sure that you write that this area in the middle is the new strands. And the ones on the outside are the template strands because we use them to make a copy. So first, here's our original. We opened it up. And once we opened it up, we added the copy of DNA. Let's go ahead and keep going on. So we have 3 different hypotheses about DNA replication. So after Franklin's data revealed how DNA is a double helix, scientists wanted to know, okay, we know the shape, but how is DNA going to be replicated? So we have 3 models that are proposed. We have the conservative model, which is this first one over here. So the conservative model is that when we make a copy of DNA, it's just a complete copy, 100%. And then we have the semi-conservative, and this is where you have DNA, and it's going to split up, and we end up making, like part of the strand is the old strand, and part of the strand is actually the new strand. Then the last one that we have is like the dispersive model. Here, let me move this little thing out of the way just so you can see this. There we go. This bottom one right here is the dispersive. So I don't know if this is hard to see, but like there's parts of like dark blue and light blue. So instead of it being like half and half, it's like sprinkled all around. So let's go ahead and and just label them right now. I know your notes don't have it. So, whoa, here we go. So go ahead and label. The first one is considered conservative. The second one is semi-conservative because it's part old and part new. And then the dispersive is like the DNA is being sprinkled. So I'll let you go ahead and write that down next to your diagram. If you can just like walk around and just make sure that they're writing this, that would be great. Because it always comes up in AP Bio. Okay. Let's go ahead and move on. So we're going to talk about what experiments did Meselson and Stahl perform. So this one is another tricky one. I saved the harder experiments for today. So they grew bacteria in a solution with a heavy isotope called N15. After several generations, they transferred the bacteria to another solution with a lighter isotope, N14. So in case you're wondering why are we using these, we use isotopes so that we can track molecules. So for example, at the way end, I can see if this is really heavy, then it's made out of N15. If this is really light, then it's made out of N14. So after the DNA replicated, they took a sample of it and then they spun it around. That's called centrifuging. Centrifuging. After another replication, then they took another sample of DNA and then they spun it around again, centrifuged it. So let's find out what ended up happening. I'm going to just pass this. So after one replication, 100% of the DNA had a medium weight. But after two replications, 50% of the DNA sample had a medium weight. And then the other 50% had a light weight. So this is actually coming back to the whole idea of, is DNA replication going to be conservative, where it's like one is going to be the original copy and the other one is going to be just the copy. And then you have semi-conservative, well, it's half of the old and half of the new. And then you have dispersive, where it's like sprinkled. The DNA is going to be sprinkled, the new and the old together. So the significance of these, though, let's go ahead and break it down. So in case you're a little bit lost, let me help you break it down. 100% of DNA after one replication had one old strand that was N15 and one new strand that was N14, which goes back to our semi-conservative model, where when I go through DNA replication, I have one strand that is the original and one that is the copy. So after two replications, 50% of DNA consisted of one old strand and one new strand. And then 50% ended up with two new strands, but it's because you end up having half of this strand, which is right here. And so it goes with another one. So in other words, it's supporting the semi-conservative model. And so this result supports that semi-conservative model of DNA replication. And so we were able to figure out, okay, when we go through DNA replication, we end up having the strand that is like part old and then part new. So I know it's kind of a lot of writing, so I'll let you go ahead and finish writing it. If I went too fast, of course, you can pause it. I hope everyone's day is going well, by the way. I can't wait to see you guys tomorrow. I'm learning a lot. Okay, let's go ahead and move on. So now what we're going to move on to is we're going to move on to the cootie catcher. So this is like those things that like, it's the fortune tellers. They're also called cootie catchers. They're the ones that like you end up folding. But today we're not cutting it. So we're going to end up cutting it tomorrow and then we'll get to review it tomorrow. You'll have 10 minutes. You need to use your chapter 16 notes from today and tomorrow. If it's completely done, you get 100. And if like one or two is missing, it's an 85. If, for example, like an entire side is missing, that's a 75. So you can think of like every single block is like worth 25 points. So in case you're wondering, I don't know how to do this, Ms. DeLeon. We're going to fold it and cut it tomorrow, but you're going to go ahead and write what the experiment is, like in as many like little words as possible. But Miss De Leon, you already had me summarize the experiments. Right. The thing is that in AP Bio, it's important to understand like how we know that DNA is the genetic material and how we know that it's the semi-conservative model versus the conservative and the dispersive. So that is why we keep going over these, because now I just gave you more experiments today and you have to know the ones from yesterday. So for example, for Griffith's mouse experiment from yesterday, you would write the experiment right here. You don't have to write anything on adenine. You don't have to write anything on the experiments. The only area that you were writing on is the part that says experiment. That's the only area, and you're just describing this as best as you can. So again, like I said, you have 10 minutes. You can only work with your shoulder partner. If you're really stuck, please text me. My number is somewhere that way. It's in green on the board. Okay, so I'll go ahead and give you 10 minutes to get this done. It will be turned in. I'll go ahead and have this up. Please go ahead and press pause so that my students can work on this and get this done. You have 10 minutes and go. Okay, the time is up now. So please make sure that your name is on it. We're not cutting it today. We'll cut it tomorrow and like do the cute foldy thing. And then we'll like use it as a study guide. It's gonna be fun. So please make sure that your name is on it. Pass it to the right. The last thing that I do have for you guys today is going to be the Candy DNA. Now we've done this in 7th grade and 9th grade, but I want to go over this just to make sure we remember the instructions, or like the description of it. So these are just some expectations for Candy DNA. Make sure you clean up any mess that you make. All toothpicks must be turned into the sub. Miss, but I want to pick my tooth. No, the last thing I want to hear is like that there's toothpicks on the floor, or like you drop it by accident, someone pokes someone. I don't want that at all. If that happens, I know they're going to end up, I don't know. I just don't want 12th grade to look bad. So just make sure all toothpicks are turned into the sub. You shouldn't be playing with it with your teeth. It needs to be turned into the sub and like thrown out. Candy can't be taken out of the classroom due to CNP policies. And also, I don't want to make a mess like in my team and any of my team members like rooms, even if like you promise to take care of it. Candy cannot be taken out of my classroom. So if you don't eat it or your partner doesn't eat it, please just like throw it away. You are not allowed to leave the room with the Ziploc bag at all. So let's go ahead and look at our Candy DNA. This is what you're making. So before I have someone pass out the little baggies, let's just review this real quick. The Twizzlers are going to be the sugar phosphate backbone. Our green marshmallows will be G's. Our pink marshmallows will be C's. Our orange marshmallows will be T's. And our yellow marshmallows will be A's. You can put them in any sequence you want. The whole point of this is just so that we can look at the Candy DNA, and then we can go ahead and see the structure, like the double helix again. So this is what we need to do. We're gonna go ahead and have, 3 people. If you can go ahead and pass out the Ziploc bags to each person, and I'll go ahead and give us a total of 5 minutes to go ahead and create this Candy DNA. So you have 5 minutes to go ahead and create it, and then we'll come back. So sub, if you can go ahead and press pause, that would be fantastic. Thank you. Okay, at this point, I should have everyone that has already made their Candy DNA. So, let's look at this real quick. So I know some of you guys are looking at it, but pause for a second. So sub, if you can just make sure that people are looking this way so we can go over this. The area of the genetic information are going to be the marshmallows. So this area right here is the marshmallows. The red licorice is not genetic information. It just holds the sides of the DNA. It's very strong. So you need to know that the red parts are the parts that stabilize the DNA to twist the way that it does. The inside is the genetic information. So this is where we have our bases. And then the outsides are going to be the sugar phosphate backbone. So with that being said, please go over this with your partner. I'll go ahead and give you 30 seconds. Wait, miss, what do I need to say again? You need to tell your partner, the marshmallows are the genetic information. The Twizzlers are the sugar phosphate backbone. If you want to twist it, you can. If you don't want to twist it, you don't have to. But please go over that with your partner. I'll go ahead and give you a total of 30 seconds, and I'll just go ahead and keep the camera rolling during the 30 seconds. Go ahead and go over it with your partner, please. Okay, time is up. So now that you have gotten to see the candy DNA, thank you for being wonderful with the sub. We need to clean up. So you need to, you need to go ahead and place all toothpicks in the Ziploc bag before you throw them out. And no, you may not take the bag with you. Any candy you don't eat or your partner doesn't eat, throw it out. We don't need to be making a mess in the senior hall. We need to keep it clean. And so once we clean up, we are done for the day. You can go ahead and work on your homework if you haven't finished it from yesterday. And that is it. Have a wonderful day. I'll see you guys tomorrow. I can't wait to do our stunted growth lab. Bye, guys.